This question relates to the study of different modes of birth on microbiomes in rats. In addition to the differences in the mode of birth between a rat that's born by C-section and vaginally, one could imagine that the C-section might impose a stress on the mother that could be vicariously experienced by the rat. So the fact that you're working with an animal model gives you opportunity to tease apart the effects of microbiome versus accompanying things. Have you looked at whether you could replicate the effect by giving a postpartum C-section to the mother, or alternatively whether you could rescue the effects of the C-section rats by swabbing them with the mother's vaginal secretions?
Yeah, no, excellent questions. So I didn't go into in great detail, but so we do control for, so we actually don't, we use a different mom, we cross-foster after C-section, so it adds another complication. So it's not a clear translational, so it's another stress. So to alleviate some of them effects, we have a control that's also cross-fostered. So that's how we're able to do. Looking at the vaginal swabbing is being done now in humans. And Gloria Dominguez-Bello is doing that in human situations. So it would be worthwhile doing in an animal model, and it's something we haven't done yet. We're looking at other interventions, and so we are testing other interventions. Changes in oxytocin, for example. We'll be presenting that at SFN next week as well, that can actually prevent some of these effects that are going on, as well as changing the microbiome itself. But yeah, that's a good suggestion.
Yes, thank you very much for your lecture and coming out here. I'm actually very excited that you're doing this research. I am actually a parent of a male born by C-section that I've recovered by 85% from autism using probiotics and a gut healing regimen. So I still have a lot of questions, and so one of my questions is, since we may think that the poor gut flora may cause immune dysregulation in these children, can you comment on what happens when they are exposed to different viruses? I've always kind of questioned what happens to their immunomodulation of viruses when they have this poor gut flora.
Sure. Well, like most things in this field, the simple answer could be I don't know, because there's so much we don't know about it. In understanding virus bacteria interactions is becoming a very hot area right now, not just in terms of inflammation like what you're suggesting, but also can we harness the effects of bacteriophages to tinker with the composition of the bacteria as well. I'm sure there are studies on barrier function in relation to chronic viral infection. I was at a meeting yesterday about HIV, and in there it's clear that there is an effect. But in terms of influenza, only studies that I'm aware of are those that would look at it in prenatal exposure to things like the flu virus, which will affect the composition of the bacteria for sure.
Okay, and just one other question. Do we think that the hormones in the women are protecting them versus the males?
Yeah, it's a great question, and we just don't know.
Okay.
And it's one of these conundrums we have in neuroscience about sex differences. Okay. And it's just interesting, and if you talk to any pediatrician or obstetrician, you know, the young boy's brain is just much more vulnerable to all types of insults for some reasons that we have no idea.
Okay, thank you very much.
That was a fascinating talk. Thank you. Towards the end, you showed some slides on effects of a probiotic on cognitive function. Could you elaborate a bit on the timeline of that action? Does it take, administration of a week, 3 to 4? You mentioned 3 to 4 weeks, but is it sustained?
Yeah. So these are just very early studies that we've done in students or university population. So is it normal in the first place? That's a whole other question. And we just gave it for 4 weeks, I think, before we did any testing. And that's we didn't look at long-term effects in terms of persistence, but it will be a very important thing to do in the future. What we do know is that in other outcomes, like in irritable bowel syndrome and in the clinical studies that have been done there with certain bacteria, bacteria act as almost like tourists. So that have an effect, but once there's a washout period, the effect can go away. So that doesn't mean they're not useful, because we know that tourists can cause a lot of collateral damage, as you can see in Washington. But it is an important thing to keep in mind, that these may be long-term changes that need to be to counteract some of the genetic influences.
Thank you.
So my lab has recently been trying to use deep sequencing to look for viruses in human brain tissue specimens. The surprise was there were tons and tons of reads that mapped to gamma proteobacter. So I thought, contaminated samples, because everybody knows the brain is sterile. But 1 minute of Googling turned up a paper by Chris Powell, and he saw a whole mess of alpha proteobacter in brain samples from people who had died of various causes. So if my discussion section says, I think I confirmed Chris Powell, will I get Rotten Tomatoes thrown at me? Or is there like a nascent sense that maybe there are actually bacteria inside the brain?
Yeah, I mean, we don't know enough. I mean, there's still a lot of skepticism about that. But the problem with some of this field is the risk of contamination and proving that it's not. And a lot of it comes from the virals field, where people have found viral things that didn't really pan out in the end. I don't know. I mean, you know what I mean? Like people have said the placenta was sterile, and now we're changing our view on that. So with technology being able to get at, and can be proven, I wouldn't rule anything out. But especially in relation to how leaky the blood-brain barrier would be at that given time.
Hi, thank you very much. I noticed in your talk you didn't talk a lot about individual or specific strains or species or taxa of bacteria, and I see in the literature very often it seems like we're trying to identify these magic bacteria that are going to fix things. And just given the diversity between people, the variability between people, but between populations, it seems that very much sort of the answer is going to come through understanding the functional eco-niches that bacteria occupy, rather than the taxonomic units to which they belong. And it just seems that for some of these problems, there are fields that are geared towards these kinds of issues. We need ecologists, we need systems biologists to help us disentangle this very complex ecosystem that we are trying to deal with. And I just wondered if you have any thoughts on that.
Yeah, I think you bring up a very important point, and really where this field is going, it is a convergence of, it needs multidisciplinary approaches to it. I focused on really just trying to share with you the concept that microbes can influence brain function and behavior, because I'm a neurobiologist, and that's where my interest is, and I collaborate with people who do the big data point of, and that's where the field is going. And you're dead right in terms of ecological niches and understanding that. What argues a little bit against it is how single bacterial strains can have such marked effects. Because they shouldn't if it was all due to a specific population-based niches. But understanding what the effects of that strain on the local ecosystem will be an important part of it. And with advances in technology and integrating big data, we will move into that direction, especially trying to parse what's going on in the immune system with what's going on in the brain.
Thank you.
Hi. So the most heavily applied industrial herbicide, at least in the United States, functions by inhibiting the shikimic acid pathway. It's the tryptophan synthesis pathway. And this is a pathway that's supposed to be safe to eat. It ends up on a lot of crops and things. It's supposed to be safe to eat because humans don't have a tryptophan, tyrosine, phenylalanine synthesis pathway. But I've seen, a lot of these microbes work in tryptophan metabolism and things like that. I guess what I'm asking is, has the concept of the essential amino acid sort of played itself out now that we've got the heritable, thousands of species and things?
That's a really interesting concept and really provocative in some ways. But when I saw, if you look at the paper from Paul Patterson and Sarkis Mazmanian, there they looked at metabolites, and a lot of these metabolites were of the tryptophan variety, and they're only bacterial. The humans could not make them without the bacteria and without the adequate substrate. So we also need to get substrate specificity. So you need to take certain things in your diet that can be acted on in these bacteria to produce these neuroactive substances. And I think the repertoire of neuroactives that we have potentially is increasing. And so I think I would agree with you in that regard. And so by taking, and that's why fibers and other dietary interventions that can be tailored in that way will produce some novel metabolites in certain novel ecosystems to be able to modulate function.
Thanks. Thank you very much.